In the previous lessons, we saw that there is a tight relationship between the structure and function of proteins. And this means that in order to understand how proteins work, we need to first understand the different aspects of their structure. And that's what we're going to do in the following lessons. But before that, I'd like to say a few words about the different ways we use to represent molecular structures in this course. And when we take all the molecules that we encounter, we can divide them into two main groups. The first one includes small molecules, for example, substrates or cofactors that are used by enzymes or other ligands. And these molecules have a small number of atoms, which allows us to show all of them and also the bonds between the different atoms. So we can see here three types of such representation. The first is called sticks, where each bond between two atoms is shown as a stick. In the second representation, each atom is shown as a sphere, and the size of the sphere depends on the van der Waals radius of each atom. We talked about it in the previous lessons. The third type of representation, called balls and sticks, combines the two previous ones, and we can see that each atom is shown as a small sphere, and which allows us to see also the bonds. And we can see that a single bond is shown as a thick stick, and a double bond is shown as two thin sticks. And we can also see that in these representations, each element is colored differently. And we can see the color scale here below. Carbon atoms are shown in green, hydrogen atoms in white, oxygens in red, nitrogens in blue, and sulfur in gold. And we have also other elements which have other colors. So this is the first type of molecules, small organic molecules. And like I said, we can show all the atoms and bonds in them. But then we have the second group, which includes macromolecules like proteins and DNA, RNA, and so on. And in this case, there is no point in showing all the atoms or all the bonds, which is what you see here, because there are so many of them that showing all of them just creates a very messy picture. So in this case, what we usually do is we use other types of representations. And the most common one is the one you see here, in which instead of showing the atoms or the bonds, we just show the chain, the backbone of the chain, in this case of a protein. And we do that by using a ribbon that goes through the entire chain and creates a cartoon of the folded state of the chain. So this is why we call this type of representation a ribbon or a cartoon representation. And it's very useful to show the general architecture of the protein. So for example, in this case, we see that the external parts of the chain contain segments that are shaped like springs. And on the other hand, the middle part contains smaller segments that are extended or stretched. And you can see that each of them is colored differently, and the stretched parts are also have a shape of an arrow, which shows the directionality of the chain. So this is a very useful way to show the general architecture of the protein, and it helps us classify proteins to different groups. And when we encounter a new protein, it also helps us to decide to which group it belongs, which also tells us usually about its function. So this is the first common type of representation when it comes to proteins. The second type is called the surface representation, because it doesn't show what happens inside a protein, it shows only the outer surface of the protein, as you can see here. And this is useful when we want to see the general structure, the general shape of the protein, but also to find binding sites. For example, we can see here such a site, These sites are easier to pinpoint in this type of representation because they appear as dents inside the structure. It also shows parts of the proteins that are exposed to the outer environment. So this is a surface representation, but usually we use a combined type of representation in which we show different elements using different types of representation and all in the same image. So in this case, we want to see the general architecture of the protein, its general shape, but we also want to see the binding site with the ligand of the protein bound to this binding site and at least some of the amino acids that bind it. So to do that, we use the ribbon representation to see the architecture. We use the semi-transparent surface representation to see the general shape of the protein. And for the ligand and amino acid that binds it, we use the balls and sticks representation. And we can also see that the amino acid, this amino acid in the binding site interacts with the ligand via a hydrogen bond, which is the dashed black line here. And this contributes to the favorable binding of the ligand to the binding site. So now we can move on and start our discussion of protein structure. When we look at the huge diversity and variety of proteins in nature, we see that they are very versatile in terms of size and shape. And we have a few examples in this image. We see, for example, proteins like insulin, which are made of a single chain and contain only a dozen or 2 dozens of amino acids. For example, insulin contains 21 amino acids. But we can also find larger and larger proteins. And we also find huge protein complexes, which are made of many different chains and may contain thousands of amino acids. A second example can be seen here. We have the protein called actin, which form large filaments or long filaments, and these filaments build the cytoskeleton inside each cell, and also the long fibers inside muscle cells, which allow these cells to contract. We can also see that the different proteins are not only different in terms of their size, but they also have different shapes, like the barrel shape here, the star shape here, and the Y shape here. So all these proteins are very different, but still all proteins in nature are built according to the same principles. And the main principle is that proteins are hierarchical, or their structure is hierarchical, which means that it contains different levels of increasing complexity. So the most basic level is what we call the primary structure, which is in fact the amino acid sequence of the protein chain. And as we saw earlier, this amino acid sequence induces the chain to fold into a specific 3-dimensional structure. And this structure is called the tertiary structure of the protein. So we have the primary and tertiary structures. We're missing the secondary structure. The secondary structure is also something that we saw earlier. It's the small segments inside the complete chain of the protein, which have, each of them has its own local fold. In this case, it has the shape of a spring. And we saw earlier that proteins may contain such segments with the shapes of springs, but also other segments that look like loops or are extended or stretched. So these are the 3 basic levels of protein structure, and they exist in virtually all proteins because all proteins contain at least one chain. Some proteins, as we saw, are made of several chains that create a complex. And when such a complex exists, the order of the chains and the way that they interact with one another is the final level of complexity, which is called the quaternary structure. So again, all proteins have the first 2 or 3 levels of complexity of the structure, and some proteins which form complexes have 4 levels of complexity: the primary, secondary, tertiary, and quaternary structure. Many proteins contain, besides the amino acids that construct their chains, also other chemical entities, which can be elemental ions, water molecules, or small organic or inorganic molecules. And we collectively refer to these as heterogroups. And this can be divided into 2 types. The first type are those that are permanently bound to the protein chain, and they're usually either metals or organic groups, which we are called prosthetic groups. And we can see an example here of the protein hemoglobin, which you probably know is responsible for binding oxygen in the lungs of animals and delivering it to the other tissues. And if we look at this structure, we can see that inside a pocket of the protein chain, there is an organic group called heme. And this is where the name of the protein comes from. This organic group contains in the middle of it an iron atom, which is responsible for binding the oxygen molecule. So this is the first type of heterogroups. The second type contains heterogroups which are transiently bound to the protein chain. And one example is the substrates of enzymes, those molecules that bind to an enzyme, convert it to product, and then leave the protein chain. Another example are molecules that act as regulators of protein activity. And a third very common example are molecules that are referred to as coenzymes or cofactors. And these are organic molecules that exist inside enzymes, and their function is to extend the catalytic capabilities of the enzyme. They carry out catalytic functions which the amino acids that compose the enzymes are either unable or they're just not good enough in carrying out. And when we look this table, when we look at these cofactors or coenzymes, we see that many of them, actually most of them, are actually chemical derivatives of vitamins. This is the list of B vitamins, and here we have vitamin C and vitamin K, all of which are the organic sources of these cofactors, these enzymatic cofactors. And if you're interested in this column, we can see the catalytic functions of each of the cofactors.